Again, welcome back, everyone. We are now moving into session 2, and this is going to be looking at analytical considerations. That's, model characterization and validation of the performance characteristics.
Good morning, everyone.
And so Graham Marsh,
welcome back
from
to day
CPath
2.
is going to be
We're going to go ahead and get started
session.
here in just a few moments.
Graham
so if everyone
joined
will
us
take a
at
seat.
CPath about 6 months ago now. And
if you're
prior
out
to
in the
that,
lobby, go ahead and
he
join
had
us
been,
in the
working in pharma, and then also at a company developing 3D models as well. So Graham, please.
All right, thanks, Nick. To talk about the analytical considerations for these complex in vitro models, we've got 2 talks lined up from Deirdre Dalmas, GSK, and Kevin Ford from the FDA. And really, we're going to hopefully use this section to talk about the ways in which we can characterize these models to get baseline data, to build confidence so that they can be applied for the appropriate context of use. Thanks, Deirdre. So I'd like to introduce Kevin Ford. He's an associate director in DARS, and he's working on their complex in vitro model characterization. So, Kevin, this is the remote control. 3 forward.
Okay, great. Thank you. Thanks, everybody. So today I'd like to highlight some of the regulatory challenges associated with validating appropriate CIVM for context of use. So the typical FDA disclaimer. So the presentation is broken down to 4 main areas. First, I'd like to highlight some of the key sources of variability in validating an in vitro model and why this is a particular challenge to regulatory agencies. Second, I'm going to focus on some of the baseline functions. In particular, I'm going to focus on urea and albumin in MPS systems. Then I'll discuss some transport and enzyme expression activity, why that's important. And lastly, I'll finish talking about how to select drugs to validate systems. So beginning with the in vitro model variability. So this slide, I know it's a bit busy, but the point is to illustrate that standardization of an in vitro model is quite complex due to the various sources of variability as illustrated here. So there are 3 main types of variability. There's biological variability, and that could include things such as differences in nutritional states or autoregulation or shear stress or anything like that that will influence the cell one way or another. So standardizing that is quite challenging. Secondly, there's device variability, and this could include something such as the type of plastic used. So for MPS systems up until now, PDMS was quite common. We're moving towards the cycloolefins, which cause drugs to adhere less to the plastic. So that's kind of where we're moving now. Another example would be flow rate, which influences the delivery of oxygen to cells. And also the type of pump, which influences flow type. So flow type could be turbulent, it could be laminar, it could be various different types. So that all influences the cells. And lastly, the manufacturing variability is broken down into 3 areas here: inter-site variability, inter-batch variability, and intra-batch variability. So that's the challenge for regulatory agencies to kind of standardize on all that variability. So it is quite a challenge, as you can appreciate, I'm sure. So one of the most common source of variability is the source of cells that are actually used in the models. So for reliable and reproducible CIVM platforms, standardization of cells used is very important. And there are 3 main types of cells, and they're not really interchangeable. There's the primary cells that are derived from organ biopsies. There's the iPSC, the induced pluripotent stem cells, that are typically derived from skin or blood. And then there's cell lines that are commercially available, which are typically derived from cancer immortalized cell lines. So you can't really use, primary cells and compare them to immortalized cells. They're very different. So standardizing the cell type is very important. So then, in addition to standardizing cell type, it's kind of obvious to say that how the cells are used is very important. And this gives you an example of, let's say, for instance, in the assessment of cellular viability, Sponsors will typically assess the cellular viability in either 2D culture or 3D culture. And each have their advantages and disadvantages. But the point here is to show that even if the same cell type is used, because they're used in this different way, it's really challenging to compare the results. So there's another source of variability. Another one, that's this long growing list now, is the temporal component, stability of the model over time. And this, these two plots here are taken from two publications from the DARS MPS lab, illustrate that CYP3A4 activity in albumin production lasts like a longer time in MPS models versus 2D and 3D models. So it's important to appreciate this because, you know, how the cells behave in a particular model as well really influences a lot. So to really get an understanding of that is very, very important. Oh, guess these things over here. Okay, moving on to baseline function. So just focusing on albumin and urea production in liver MPS, these are the two most common biomarkers of liver in general cell health. And these biomarkers are used because they cover a wide range of cellular organelle functionality and can offer a view into the overall health status of the hepatocyte. So just looking at the biosynthetic pathway of albumin here, you see that there are parts that occur in the, the liver, the rough endoplasmic reticulum, and the Golgi apparatus. So you get a kind of an appreciation of how the drug is affecting those three areas by just measuring albumin. And similarly, by measuring urea, which is produced from ammonia in the urea cycle, two of the steps which occur in the mitochondria, so that will give you an indication of mitochondrial health. So very, very easy biomarkers to measure, and they are quite informative. So it's important to also realize that, you know, albumin production and urea synthesis should be measured in the initial assessment of liver MPS so that the model can be optimized to reach the desired fundamental target thresholds at an early stage. And this is taken from that body paper, which I'm sure you're all familiar with. And in this paper, they show that, you know, they define kind of like strict thresholds for various endpoints. So for instance, they say here that for albumin, it should be between 37 and 105, urea to be 56 to 159. However, some of the major MPS producers, which are many of whom are here today, such as Emulate and CMBio, actually show that there's the number can be less. So it's important to realize how your cells interact with your device and not necessarily just rely on one threshold that published in the literature that kind of covers everything. One size really doesn't fit all. So moving on to examination of the transporter and enzyme expression activity. So looking at the transporters, so in that same paper and two additional papers from the IQ-MPS consortium, they show that various transporters should be verified for the presence of these transporters by gene expression prior to running any investigation. And for the liver, because that's what we're focusing on today, they just mentioned three transporters. And I just want to make the point here that it really depends on the context of use. So for instance, I'm sure you're all familiar, for filing INDs, you need to focus on additional transporters for DDI studies, drug-drug interaction studies, including here. So the ones I highlight in red there are additional ones that should be measured using gene expression if you're using your MPS system for drug-drug interactions. So it's important to just go, sometimes you're going to need to go beyond what they mentioned in that paper. So looking at the enzymes recommended for screening by the IQ-MPS consortium. So it's important to verify that, you know, CYPs are present, but by doing the gene expression, it's actually informing you if a particular enzyme is absent. And this is important because certain autosomal recessive conditions, various CYPs are actually missing. So for instance, P450 oxoreductase deficiency, various CYPs are just not expressed by patients. So it's important to know where your cells are coming from. So depending on the context of use, knowing if polymorphisms or SIPs are present is critical. And the obvious example here is 2D6. So patients can range, or population can range all the way from poor metabolizers all the way up to ultra rapid metabolizers, depending on the gene copy. So it's really very diverse there. So really get a handle of that early on is very important. And then it's important also just to mention, I'm sure you all know this, but not just to rely on gene expression data, to actually perform confirmatory functional CYP activity assays is important, because oftentimes the expression can be misleading. So there are quite a few CYPs that they recommend here, as listed here. I'm sure you're all familiar with them, and they're actually quite good ones. So the last part of my presentation, I'm going to discuss the selection of drugs to validate systems. So this, again, is from that body paper. They present a list of 20 chemicals, including 1 oligonucleotide, 13 DILI-causing drugs, and 7 that do not cause DILI. And they say apply this compound list for evaluation of MPS sensitivity, in yellow there. However, we've identified in DARS some concerns with this list. First of all, there's a lack of structural diversity. So a structural analysis of 254 DILI severe drugs reported to FDA reveals that the IQ-MPS test set is actually missing 8 out of 12 of the most common DILI-associated toxicophores, and I list them there in red. So it doesn't have good coverage of even the most severe DILI. Secondly, the negative controls, several drugs described as having no DILI concerns were actually found to demonstrate mild to moderate DILI effects. Thirdly, several of the drugs are not commercially available and require custom synthesis. So this is quite onerous for sponsors. So I don't think this is how it should be done. Fourth, salts were not specified. So certain salts may be associated with increased risk of DILI. And the classic example of this is mesolates, right? So any alkyl mesylate, they're classic mutagens and they'll cause hepatotoxicity. So you want to just be careful of that. Keep an eye on the salt also, not just the active drug substance. So lastly, the importance of high purity is not mentioned. And this is very important because of genotoxic impurity concerns. So again, it's really important to highlight that, even though it might be a basic thing, but it's important to highlight it. So in light of these concerns, we decided in DARS to probe structural and chemical diversity and see if we could improve on this training set of drugs. So the first step we did is we performed a molecular descriptor analysis of the drugs in this data set called DILI rank that's actually publicly available on the FDA website. And they break up the drugs into severe, moderate, mild, and negative in terms of DILI effects. And there's quite a few in each bucket, like at least 200 plus drugs. So we analyzed the key drivers of molecular descriptors associated with those endpoints. And then once we had that data, we reviewed literature data, clinical reports, and the FAERS data, which is FDA Adverse Event Reporting System, for each of the drugs. So using that data, we performed principal component analysis of the phys-chem properties known to be associated with DILI. And as you can see here, one of the most well-known drivers of DILI is the number of aromatic rings, which is the N-aromatic down here. So when you plot this, the C log P, which is a measure of lipophilicity, versus the N-aromatic, what you'll notice is the drugs from the liver MPS consortium actually don't contain any of the drugs in this 0 and the 4 and the 5. And it's concerning because the 0 is where most of the drugs lie. So there's no coverage whatsoever there. So it's important that we do get coverage there. And so not to tease you guys too much, but we have actually come up with a list, a comprehensive list with all of those parameters in mind. And we've decided that it would be best to drop the drugs in red for various reasons that we will provide in a publication on this area from DARS soon. So actually with that, I'd like to thank everybody listed here, including most particularly members of the MPS lab in DARS. and also David Strauss and Rod Rouse here, and everybody else listed here. It takes a village to produce data like this, so thank you, everybody.